Everybody's been posting at the hashtag #RUTalkingScience, and before we begin the second half, I want to acknowledge the winner of today's Talking Science social media post competition. This post, I think, is really fun. It certainly represents some really cool aspects. Both there's a little artistic interpretation, and I think that we can pull a little genetic study into it as well. So this is why it was chosen. So the winner of the social media competition is like a little drum roll, right? Because you get this awesome canteen. Yeah. Yuri Han, oh, come on down for your water bottle. Thanks. There you go. Oh, that's good. Wait, who's the mosquito now? Oh, you and you're not? Yeah, I'm like, cool. Awesome. So congrats to that. That's really fun. I love it. So I'm now excited to introduce you to another fantastic scientist, Dr. Ben Matthews. Ben is a research specialist in the Vassal Laboratory, where he studies genes that are critical to the life cycle and behavior of the Aedes aegypti mosquito. The Aedes aegypti uses its senses of smell and taste to hunt and feed on human blood, which, as you have learned, the females must do in order to lay eggs. As a result of their blood-seeking activity, these mosquitoes transmit diseases such as yellow fever, dengue, and Zika to hundreds of millions of people worldwide, often with life-threatening effects. This research rose to critical importance in 2016 as the Zika crisis intensified. Ben will tell us about his efforts to engineer mosquitoes that would be less attracted to human hosts. In particular, he will talk about the use of these amazing molecular scissors, CRISPR, a breakthrough through molecular technology that enables scientists to make precise edits in the genome of any organism. So CRISPR is a really fantastic tool that we borrowed from ancient bacteria. Ben earned his undergraduate degree at the California Institute of Technology and his PhD at Columbia University. He received a 2010 Henry and Marie Josie Kravis Fellowship and a 2011 Jane Coffin Childs Fellowship. So without further ado, I'd like to welcome Ben to the podium, who will continue his talk on cut and paste genomes using CRISPR to stop mosquitoes. So thank you, Ben.
Thanks. All right, can you hear me? Yep. So thanks all for sticking through, no problem. Yeah. I'll trade coffee, water. Thank you all for sticking around for the afternoon. It's great to have you all here in the cold. And so, as Jeannie said, I'm going to talk about some of the gene editing work that we do in the mosquito that you learned about this morning from Leslie. So I've been in Leslie's lab for about 7 years now, and I work on tools that we develop in order to edit the genome of the mosquito to do a lot of different things, both to alter their attraction to human hosts, but also to figure out how they decide where they're going to lay their eggs. And so I won't talk too much about the biology of that in particular, but just know that these tools can be applied to many of the questions that Leslie raised, that I study in the day-to-day working in the lab, and also a lot of the great ideas that you guys came up with in the Q&A earlier on. So CRISPR is applicable to mosquitoes, to, as Jeannie said, many, many, many organisms now, including humans. And so I hope to talk a little bit about the basics of it, and then we'll get into some of the specifics in mosquitoes, and then maybe a little bit broader as well. So this is just to make sure we're all back on the same page, even though it was hard to miss during lunch. But this is a female mosquito full of blood. And the Aedes aegypti mosquito that we study is the vector for arboviruses like Zika, chikungunya, dengue. And so this is actually from one of the mosquitoes in our lab that has been genetically modified. And so you can't tell at this stage, and I'll talk a little bit about how we did it, and then what that looks like at the molecular level. All right, so this is slightly different from the map that Leslie showed earlier, but I think it reemphasizes the point that wherever the mosquito goes, disease follows. And wherever we go, the mosquito follows until the winters get too cold that they can't overwinter. And so this is a map of not dengue fever or yellow fever or any of the other diseases we've talked about, but of the mosquito itself. And so this is about 2 years old now, and you can notice that in the US, especially on the eastern seaboard and in parts of California, there is this kind of creeping northward spread of the geographical distribution of the mosquito. And so as the climate warms, as winters get a little bit warmer, or as the travel on trains and trucks and planes and things like that get more frequent, we are going to have these mosquitoes in places that they weren't before. And it's not on this map, but I will also just point out that 2 days ago I was in New Zealand, which is somewhere off the coast here, which is just opposite of Queensland, Australia, which Leslie talked about before. And dengue is absolutely endemic in Queensland, and it's a huge problem. And if we were to see New Zealand here, it would be bright blue. And that's because it's a small island, and they can actually be pretty effective at the biosecurity, they call it. They basically stop every shipment at the border and look for the presence of things like mosquito larvae and stuff like that. So if you're on an island, you can do all right, but for the rest of the world where the climate is habitable for these mosquitoes, you're going to find them. All right, and this is just zooming in a little bit, and we'll talk about some of the efforts to use genetically modified mosquitoes, especially in Brazil during the Olympics and the World Cup in the past few years. There have actually been targeted release programs of genetically modified mosquitoes in areas that have high risk of dengue and yellow fever. And we'll talk about some of the efforts to do that as well. All right, so this is from right around World War II. This is Jones Beach. Anybody been to Jones Beach? Yeah, exactly. So what you're seeing here is a truck driving on Jones Beach, spraying a fog of insecticide across the beach. And so we used to do this on a very regular basis, and it's actually one of the reasons that we don't have dengue fever or yellow fever in the States anymore, is that we got very good at using chemicals to control mosquitoes. But as you can tell from your reactions, this is not something that we tend to think about doing these days, but we still do it. So there are parts of New York City even that will have aerial spray of insecticides every summer. Does anybody live in a neighborhood where this happens? Yeah, we got one. Where do you live?
Queens.
Yeah, Queens. So in Queens, they'll send out emails, they'll send out flyers, they'll say, don't let your kids go to the playground on this night because we're going to fly helicopters or drones now and actually dump insecticide. And so they're trying to kill larvae and they're trying to kill adults. And so this is how we keep down mosquito populations in parts of the world where you can get away with it. But obviously, we don't want to think about this as a way of controlling mosquitoes long term. And even if we could, if we just go back to the geographical distribution, it's just not possible to use chemicals to control mosquitoes everywhere. So we may be able to use chemicals at a local level, even if they don't evolve the resistance to the specific chemicals that we use, like Leslie talked about earlier. So this is kind of the state of the art still in a lot of ways for how we control mosquitoes. All right, but we're going to talk more about the genetics today. So before we get into the tools, I just wanted to make sure we're all on the same page. So I'm sorry if this is, this is probably 8th grade, 7th grade, 6th grade biology, but just so that we go over some of the terms. So the central dogma of molecular biology is that information is encoded in DNA, which is a double helical structure like this, and there are individual base pairs that have an affinity for one another. So you have your A's and your T's and your G's and your C's. And then this is encoded in strands of double helical structures like that. And then there's a process called transcription where molecular machines kind of travel along this DNA and they convert it into RNA. And so we're going to come back to RNA later on when we talk about CRISPR, but one of the important things to know about RNA is that it has a very similar structure to DNA, and that there's actually, even though they swap a T, so they swap the thymine in DNA for a U, a uracil, so now we have orange bases instead of red, there is still this base pair affinity, so that not only can DNA bind to DNA, but DNA can bind to RNA using a very simple rule, similar to the way that DNA binding happens. And so this is something that CRISPR exploits, and one of the reasons it's become so popular these days. And so then finally, we have a strand of RNA. It's a single-stranded molecule. Typically, if you're going to produce a protein, it's called messenger RNA or mRNA. And then that gets translated by another set of molecular machines, enzymes and ribosomes and all the like, to produce proteins. And so proteins are what we think about when we think about the building blocks of biological activity within a cell. They can be on the membrane, they can be receptors, or they can be within the cell, each doing their own job. And so this is kind of the flow of information in all organisms that we study today. So we'll come back to this. And then one more term is just genome. And genome just refers to the entire set of instructions for a cell, basically. So every cell in our body has 23 pairs of chromosomes that all have a complete copy of our genome with small mutations from cell to cell in some cases. And this is true for any organism. So a mosquito has a genome that's present in all of its cells, and that's what we're going to talk about editing when we edit genomes. All right, so this is just to see where we are in terms of scale, right? So I just want to see a show of hands. We're going to go down the list, and I'm asking you how big the human genome is on average. And so we have 4 choices. I'll give you the first set of numbers. It's 3.2. What you have to give me is the scale. So we're orders of magnitude here. So the first choice is going to be 3.2 thousand, and the last choice is going to be 3.2 trillion. So let's see a show of hands for A, who thinks our genome is 3.2 thousand. All right. B? C? And D. All right, so we have some Ds, a lot of Cs, no As, and no Bs. So I'll basically walk you through. These two are not correct. So our genome is much bigger than this. And it's not 3.2 trillion, it's 3.2 billion. So good job to all of you who are in the right order of magnitude. Like that, this is most of what science is trying to get us to the right order of magnitude. So we're talking billions of letters and not trillions of letters. All right, so a mosquito is about 240-fold smaller than a human being. So how big do you think the mosquito genome is? Anybody? All right, I'll give you the same set of instructions. I heard 1.3 million. So again, I'm going to give you the first number. So it's 1.3, and is it 1,000, million, billion, or trillion? So what about A? Anybody think it's 1.3 thousand? 1. B? All right, we got some more. C? And D. All right, so no Ds this time, maybe 1 or 2 Ds. We got a few more Bs, which would make sense because if the animal is about 240-fold smaller, the genome might be a little bit smaller. But I'm actually going to cut that one out early. So a mosquito has roughly the same size genome as a human being, right? So this is kind of incredible when you think about it. And actually, if you talk about the smallest genome in known organisms, it sort of makes sense. It's a virus. And it has thousands of bases, not millions, billions, or trillions. So a very small virus has a very small genome. But the biggest genome that we know of is actually in an amoeba. And that's hundreds and hundreds of billions of bases. And so an amoeba is a small protozoan, and the genome is massively inflated compared to ours or to a mosquito's. So the size of the animal doesn't have an influence really on the size of its genome. And there are a lot of different reasons for that, but just so you're aware that whether we're talking about editing a bacterium or a mosquito or a human, we're dealing with billions of base pairs of DNA typically, if not hundreds of millions. All right, so sequencing genomes is something that has kind of come about in my scientific lifetime and should be something that you never really have to think about whether it's a possibility or not as you go forward in science, because it is, and it's increasingly getting cheaper. They sell devices now that fit in the palm of your hand that can sequence the genome of any material that you collect in the field. So you can go out and collect samples from the field, and you can sequence a genome on a small portable DNA sequencer in hours in some cases. But that has not always been true. So to sequence the first human genome, it cost about $3 billion and took 10 years. And it was a collaboration between the US government, some other foreign governments, and commercial enterprises that were developing the tools and the technology to actually read out the genome of a human being. And it took about 10 years, and it finished in 2000 with the publication of what we call a draft genome, which was about 90% complete. So at that point, we were still missing 10% of the genome. Today, we can do that for about $1,000 in just a few days. And so this is just in the last 15 or 17 years, the price and the speed at which we can sequence genomes has come way, way down. And so consequently now, when you go to the doctor, if you have certain types of cancer, one of the first things that they'll do is they'll actually sequence the genome of those cancer cells to figure out the best treatment going forward. And that's going to become more and more prevalent as we continue to develop cheaper and faster tools to do this. And so reading a human genome, we all have differences in our genomes, and that's going to tell us a little bit about the phenotypes that we display, whether it's hair color or height or something like that. But it can also tell you, if you look in certain cells, especially cancerous cells, what's gone wrong in those cells so you can develop the best treatment as we go forward. And so this is something called personalized medicine, which you're going to hear more and more about And so that's why we would want to sequence all of our genomes, because we're all a little bit different from one another. But the other thing that we can do with this technology now is we can read the genome of very diverse species, like the mosquito. And so if it took $3 billion in 10 years to sequence the genome of the mosquito, we may not do it, because that's a huge investment of time and effort, and there are many, many, many thousands of species around the world that we would want the genomes of, and so you'd have to kind of fight for which we're going to get their genome sequenced. But now that it's so cheap, we do this on a routine basis, and we can start to compare the genomes of different mosquitoes, different insects, and all sorts of different animals. And this is helping us learn about evolution, both on the level of single genes, because we share a lot of the same genes between humans and mosquitoes, but also about genomes and why they make certain animals better at certain things. So you could compare the malaria mosquito to the dengue fever mosquito, for example. And if you have both of those genomes, you can immediately kind of see the differences and start to make predictions about why one animal has certain phenotypes and the other has different phenotypes. And so what Leslie said is very true, that 2 years ago now, we started an effort kind of piggybacking on both the public interest, but also the influx of money that went into studying the vector for Zika virus and the virus itself. And we decided to resequence the Aedes aegypti genome. And so this is on the front page of the New York Times from an article by Amy Harmon, which was fantastic. But this was the state of the Aedes aegypti genome before we started. And so every one of these pictures, these little squiggly lines you're seeing, is a piece of consecutive DNA. So it's all those letters, those billions of letters, put together into the biggest pieces that we could at the time. And so up until we did our new genome, we had thousands of pieces, and they were all very small. But the actual mosquito genome only lies on 3 chromosomes. So instead of these thousands of pieces, what we wanted to do is complete it and get it down to the 3 chromosomes so we could read them end to end and say, this is actually what the mosquito genome looks like. And so it took not 10 years and not billions of dollars, but about 2 years and 65, 70 people. So it's still a very collaborative thing, but it's now very, very possible to sequence even large and repetitive genomes with very little effort compared to the original human genomes. And so this, like Leslie said, just came out last week, and so we hope that this is going to be very helpful going forward, not just to our work, but to others who study the mosquito. And I'll try and make the case for you that in order to edit genomes, you first have to understand what the genome is that you're trying to edit. And so if it's the human genome and you're trying to manipulate a certain gene or a certain protein, you don't want to have unintended side effects. So you need to know what all of the other sequences are as well, so you can avoid targeting those with the reagents that you're trying to build. And so we hope that this is going to do similar things for the mosquito going forward. All right. So typically, even when I did my PhD about 10 years ago now, if you wanted to study genetics in the context of animal behavior, you really had kind of 4 options, right? So you could study humans and you could sequence their genomes, but you can't do experiments with human genetics. You just can't do it. And it's becoming potentially possible, but it's still not going to be the type of experiment where you come up with a hypothesis. You say, I want to take a dopamine receptor in your brain. I predict that if I mutate it, you're going to have this particular behavioral phenotype. Nobody is ever going to let you do that experiment, even if it were possible. So we would turn to one of these model organisms. So we have a mouse, we have a zebrafish, we have a fruit fly, Drosophila melanogaster, and anybody know what this one is? Yeah. C. elegans. Well done. So C. elegans is a tiny, tiny worm. These are not to scale, so this is not a giant mouse-sized worm. But it's a tiny, tiny worm that has only 300 or so neurons in its brain, or the nervous system of the animal. And so we can study it because it's an extremely reduced complexity in terms of the number of cells in the nervous system and the wiring between them. And so these were kind of the options. But if you want to study a mosquito, which one of these might you pick? If you were really interested in a mosquito and 10 years ago you came to a lab and you said, I want to study the genetics of what makes a mosquito find and bite people, which of those would you choose? Yeah, you would probably choose the fruit fly, right? Because it looks similar, it's got wings, it's got 6 legs, it's arthropidic. So if you look at the phylogeny of evolution, this is actually going to be the closest to a mosquito of all of these. But what's one of the downsides? What can we not study in a fruit fly that we might want to study in a mosquito? Yeah. Exactly, the desire to bite humans, right? A fruit fly does not care about us one way or the other. And in fact, it would be dangerous if they were always flying around and trying to target skin. We'd be swatting them and biting them and trying to kill them with insecticide just the same way we do with mosquitoes. So we can't study the desire for human blood. We also can't study the anatomy that it would take for them to bite human skin because they just don't do it. They have rotting fruit as their meal, so they don't have to pierce, they don't have to suck, they don't have to do all of that. And they also can't harbor the type of viral disease that mosquitoes can. So if you cared about why mosquitoes transmit dengue, you can try really, really hard to get a fruit fly to be infected with dengue, but you can't do it. So there's just a whole wealth of scientific questions that you can't address with these model organisms. And I would argue that that's true in human health as well. So if you look at the history of developing drugs against cancer, for the longest time we relied on mice to try and develop drugs that would target human cancers. And so you would give a mouse cancer, we're very good at that. And then we're also very good at developing drugs that can treat cancer in mice. So we're much, much better at treating cancers, some types of cancers in mice than we are in humans. And when you try and take those drugs from mice to humans, a lot of times they just don't work because there are limits to what you can learn from studying the biology of one species and then trying to apply it to another. So it's becoming more and more important not just to understand the genomes of humans and of disease vectors like Aedes aegypti, but also any animal that has interesting biology. If you want to understand the genetics of that organism, you really have to dig down into the genetics of that organism itself and not rely on one of these 4 models. And so the concept of a model organism is still around. So if you look at the scientific literature, the vast majority of papers published are going to be on one of these 4 models, at least in terms of behavior. But we're getting into a world now where that doesn't have to be the case, and increasingly, it's not the case, where if we want to study the mosquito in terms of its genetics, we're actually going to do the genetics in the mosquito. And so that's kind of what we're going to talk about for the rest of today. All right. So sequencing genomes is great, and it's fantastic that we can do it. It's absolutely essential to be able to start to study genetics. But what it really lets you do is read the genome, all 3 billion or 1.3 billion base pairs end to end, and then we can just basically think, right? So we read it, we align it, we can manipulate it on a computer, we can predict the types of proteins that it encodes. But at the end of the day, all we can really do is make predictions about what specific genes do and why they look the way they do in a mosquito compared to a fruit fly or a mosquito compared to a human. But to actually test the kind of scientific validity of these hypotheses, we need to actually do an experiment, right? So this is the scientific process. You don't just sit around and do thought experiments all day, unless you're a theoretical physicist, in which case that's pretty much what you do. But if you're a biologist especially, you need to design well-controlled, carefully thought-out experiments and then test your ideas, test your hypotheses. And so if we're doing genetics, what do we need to test a hypothesis? We need a way to make changes to the genomes of the animals that we're studying so that we can see what the effect is on the animal, on the behavior of the animal, on the biology of its disease transmission, whatever the question is you want to study. We need to be able to edit the DNA and then do the experiment to ask what happened to the organism. And so ideally, we want the equivalent of a word processor, right? So we have our 3 billion letters from end to end, and I want to be able to sit at a computer and say, I want this word to move from here to there, and I want to take this word and delete it, and I want to make very precise changes so that maybe we make the gene that we're interested in the dengue fever mosquito, maybe we make it look like the same gene in the malaria mosquito, and then we ask what happens when we make that change. And so you can basically imagine the experiment, and if you have this tool to make the precise edit to the genome, then you can ask what happens after you've done that experiment. All right, so this is what you'll see if you open a magazine. So this is what Wired magazine would have you think that I do every day. So I'm pulling DNA out from end to end. I scan up and down, get some tweezers maybe for good effect, and then I just kind of pull out a spot, and then I move it up and down. And so this is not what I do every day. Unfortunately, it looks kind of fun. This is also not what I do every day. But I think the analogy is pretty good, right? So just like a word processor has all of the functions that you would want to be able to edit the DNA, the very first thing we have to do, because DNA is in this double helix form, right? And so the double helix is good for all sorts of things and really is the reason that we're here and as evolved as we are, because it contains 2 copies of everything, right? So it has 2 strands of DNA. They're self-complementary. So if you lose one strand, you can completely recreate the information in the double-stranded form from the other strand. And so that's why we are actually fairly tolerant of a lot of different types of mutations because we have, in every cell, we have 2 copies of every single base pair on every single chromosome. So the first thing that we have to do in order to make a change to the double-stranded form is we actually have to make a break. So we call it a double-stranded DNA break because we need to be able to cut the DNA strand at the specific part that we're interested in, and then we can make our change. So that's the very first thing that we have to do. And so the concept of a pair of molecular scissors is actually not that far off from reality. But I will say they don't look anything like this. This is a very kind of human-centric view of what we're doing. But the concept is there. We need a pair of molecular scissors to actually make the break that we need to make. All right, so this is kind of the process, right? We have our scissors, and then we need a second domain on this little machine that are going to target the scissors only to one particular spot in the genome. Because the other thing that we need to do is ensure that it's precise. I might want to make a cut in the genome at one spot, but if at the same time I'm making the cut here, I'm making 400 other cuts all over the genome, that's not going to be good. Not only for the animal, it's probably not going to survive, but also because then I can't do the properly controlled experiment where I have only made the one edit that I want to make. Now I'm trying to figure out whether, was it the one thing that I did here, or was it the 300 other things that I did that's actually causing the phenotype to change? So we need a way to target these scissors to a very particular spot of DNA. And so nature has pretty much done this for us in a lot of different ways. And so about 20 years ago, people started thinking about using these proteins called restriction enzymes, which are basically proteins. So in this diagram, it's this green kind of coiled machine here that actually has a structure that allows it to bind inside the grooves of the double helix of DNA. And so these things have evolved in nature so that they can target very specific sequences of DNA. So this is great. This is pretty much what we need. But we have to come up with a way to kind of, instead of what this protein normally does, which is probably not to make a double-strand break, but to start transcription or do something else at the specific sequence, we have to couple it to the scissors. And so it turns out that you can basically do exactly that. And so this is just an older citation, but it's been extremely influential in the field of genome editing because it kind of came up with the concept of what people call chimeric restriction enzymes or restriction endonucleases. And so chimera just means you take these two things, you jam them together, you probably have a little linker so that they don't interfere with each other, and then you just set them loose. And if you do it in the right way, you can actually engineer a protein that can target a specific sequence of DNA, and then once that's happened, the scissors come in, make their cut, and then everything goes away. And so it's great on paper, and it actually works really well in a limited set of circumstances. It just turns out it's difficult to engineer these proteins. And so we'll come back to that in a second. But I did want to tell you that CRISPR is a tool that has been developed in the past 5 or 6 years that makes our job a lot easier. But the job, we've already known what the job was, and for 20, 25 years now, we've had other tools that can do a very similar thing. They're just a little bit harder to work with. All right, so this was the history of the first kind of set of chimeric endonucleases. So the first ones, which we were actually using in Leslie's lab when I arrived 7 years ago, because that was in the pre-CRISPR era, which it really is just the last 5 years now, we were using these things called zinc finger nucleases, which have a protein domain that finds the right sequence of DNA and then a nuclease that cuts. And then in 2010, everybody got really excited because there was this new protein interface that we actually found from plants and from viruses that infect plants called tail nucleases or talons that were a little bit more flexible to work with. And so zinc fingers we had to buy from a company. And so if we wanted to target one gene, well, how much was the first one? $25,000 to target one gene. And so we have tens of thousands of genes in our genome. We have probably 18, 20,000 genes in the human genome. Mosquitoes have 12,000, 13, 14 maybe. So if we actually wanted to do anything on a scale bigger than 1 or 2 or 3 genes, the money was going to add up very quickly, and it also took months for them to build them and develop them and test them and all of that. So talons, we got really excited because we're like, okay, now we can do this at the bench. And now instead of $25,000, it's going to cost us $1,000. We're going to do it in a few weeks, maybe, if we're lucky, if we're good, and then we're going to go to town. And so there was a brief 2-year period when everybody got really excited about talons, and then in 2012, CRISPR came around, and you won't hear anybody talking about talons anymore. So it was kind of this unfortunate period where people had 15 years to play with zinc fingers and get them working just the way they wanted them. And there are a lot of clinical tools, so tools to treat human disease that are actually based on zinc fingers that are in the clinic now, and some of them are still being tested. And so I want to make it very clear that zinc fingers are not dead because we have CRISPR. And we'll talk about CRISPR in a second, but I want to give zinc fingers their due. They work very well. And once you have them, especially in the human context, and they've been tested and gone through all of the drug pipelines, if they work, and they're absolutely still functional. But that said, if you were doing genetics today, and you had a boss who told you to start by making zinc fingers, you should find another lab, and you should start over. Because if you were trying to do something that would require you to target several genes, or dozens of genes, or even hundreds of genes, some people do it genome-wide in humans, so they're going to target all 18,000 human genes, you want to use CRISPR. And so the one thing that you need to know about what makes CRISPR different from the tools that came before it, and if you remember nothing else from the next part of this, it's that it's RNA-guided rather than protein-guided. So it essentially still has the molecular scissors. It's still got that domain that cuts the DNA. But what's fundamentally different about these is how it finds the precise sequence that it wants to target. And so rather than designing a protein, which is this big three-dimensional blob that has to target the specific grooves of individual bases, CRISPR-Cas systems use RNA. And the reason that that's so much more flexible is because they have this really simple rule, right? So it's the base pairings, the same way that DNA binds to DNA, DNA also binds to RNA. So if it's just like making kind of a negative image of the DNA. So if you want to target a specific sequence of DNA, you just have to design a short RNA molecule that's the exact inverse of that DNA. And so you can do it at the bench in hours rather than many months and many tens of thousands of dollars. All right, so that doesn't actually tell you what CRISPR is, and so I'm going to talk about it a little bit. First of all, it's an extremely clever acronym, right? So if they had switched around any of these words and we were trying to talk about CRISPR or CRISPR, we wouldn't be having this conversation, right? The fact that we can say CRISPR is probably one of the most marketable parts of this tool. But that said, each of these words does describe something about what's going on. And so it stands for clustered, regularly interspersed, short palindromic repeats. Right? Right. And then Cas is the other part, so you'll often see CRISPR-Cas systems, or CRISPR-Cas9 even, and we'll talk about that. And Cas just stands for CRISPR-associated system. All right, so we're done. That's it. That's all you need to know about CRISPR, is that this is what the acronyms are, and it uses RNA to target rather than proteins. But that said, it's kind of a beautiful biological study in its own right, and one of the pioneers of this is a woman at UC Berkeley named Jennifer Doudna, and she gave a talk in this room that was packed to the gills. I think I was standing out by the second exit sign in violation of all the New York City fire codes, because she came to give a talk about how her early work on CRISPR kind of led to the tools that everybody who studies genetics is using today. And what she said is that she sent an application to the NIH about this 10 years ago, 15 years ago maybe, and it was rejected. And it was rejected because the comments came back and they said, we don't see any potential for the use of this crazy little bacterial immune system in human health, so we're not going to give you money to study it. And so what she did then is she went to the National Science Foundation, the NSF, and they said, sure, sounds interesting. It's just a quirky little biological problem. At the time, they had no idea that it was going to become so powerful as a tool, but they just were following up on a really kind of interesting biological question that they had found by sequencing the genome of lots of different bacteria. And so you need to find kind of forward-thinking funding agencies. So Howard Hughes, which funds our work, and Bill and Melinda Gates is another one. So you have to find people who are willing to fund quirky little biological questions. And they don't have to go to the lab and be like, I'm going to cure cancer by next Tuesday, and I need $1 billion to do it. Let's go. If that were the only problem, we'd be done. So there's still a big place for basic science and basic science funding in the development of all these new tools. And so this is just an extremely simplified view. It comes from the lab of Luciano Marafini, who's actually here at Rockefeller, and was another one of the early pioneers studying CRISPR in bacteria. So he's studying it in the organisms in which it evolved. And so that's important to know, is that CRISPR wasn't invented. We didn't come up with it wholesale. We actually just stole it from bacteria and are now applying it to mosquitoes and humans and all the other animals that we're studying. And so it's basically, at its core, it's an immune system for bacteria, right? So our immune system, what does it do? When we're first exposed to a disease, our body reacts and tries to fight that with the best tools that it has, but it may not know how to fight a certain virus or a certain bacterium or something else. So what it does is, in addition to kind of generally trying to fight an infection, it's busy, our immune system is busy generating very specific antibodies so that the next time we're exposed to something, we're not going to become ill. So that's chickenpox, right? So the first time you get chickenpox, you get the phenotype. You get the pox, you get the red marks. It knocks you out for a few days. But then the next time you're exposed, your body has been so good at developing antibodies for chickenpox that you now are immune, essentially. And so it turns out bacteria have the same problem. They're small unicellular things, but they are also being attacked by things like bacteriophages, which look like this, which is an RNA virus in a lot of cases. So it's a virus that tries to infect bacteria. And so what it does is it actually will inject its genome into the bacteria and then try and trick the bacteria into producing the proteins that it wants the bacteria to produce rather than the proteins that the bacteria itself would normally produce. And so it's this really clever kind of parasitic mechanism that these phages have come up with to try and influence bacteria and take them over and use them as a way to replicate the phage and not the bacteria itself. So that, first off, is extremely cool. And what's even cooler is that the bacteria realized that they had to fight back, or evolution developed a mechanism by which they could fight back, and that's really where CRISPR came from. And so rather than developing antibodies in the way that we do, what bacteria have learned how to do is take this genome that the phage has injected into them and just chop it up really fine into little pieces. So usually about 20 base pairs, 23 base pairs. And then that becomes a memory of the phage that has already infected them. So it will take the phage the first time, and it'll be like, I don't know what you are, but you're foreign, and I don't like you, so I'm just going to chop you up into 20 base pair little pieces. But then I'm going to take some of those 20 base pair pieces and put them in my own genome. So me, the bacteria, am going to take these small pieces of the viral genome and actually insert them into these arrays. So these are the short, regularly interspersed palindromic repeats that people had first picked up on when they were sequencing bacteria. And they're like, that's funny, that doesn't look like a protein coding gene or anything else that we know. What it was is it was actually this kind of molecular memory of the phages that that bacterium had been exposed to. And so this becomes their antibody system. And so what they do is once they're encoded in the genome, they then will go through a process by which they make RNAs, these short CRISPR RNAs, from these arrays. And so the business end of this CRISPR RNA is that original sequence, which is complementary to the phage genome. So then when the next time the phage tries to attack that bacterium, it has all of these circulating CRISPR RNAs. And because RNA binds DNA perfectly, you can predict how that binding is going to happen. All this CRISPR RNA has to do is bind the phage genome and then bring along a friend. And so that friend is the Cas protein. It's that blue blob in this cartoon, and we'll talk about it later. But if you had to guess what this blue blob was going to do, what would you think? Anybody? Exactly. This is the molecular scissors right here. It's going to cut the DNA. So the RNA that the bacterium produces recognizes the phage, because that's where it came from in the first place, right? It's just a template that was generated from the phage. It's going to bind, then it's going to bring over its buddy, this big pair of molecular scissors, and then before the phage can do anything at all, it's going to get cut up. And so it's going to get cut up before it can actually make the bacteria produce any of its proteins. The infection goes away, and the bacterium is very happy and healthy and lives to fight off another infection tomorrow, or in 10 minutes. So this is really the basis of the CRISPR system. So we have taken the immune system of bacteria and archaea and these really old, kind of evolutionarily simple, as we like to think of them, organisms, and we've been like, that's extremely clever and simple. We're basically going to copy your mechanism and your pair of molecular scissors, and we're going to introduce them into mosquitoes or humans or mice or whatever it is that we want to study. Because it's so simple, it just might work. So that was the insight that people had about 5 years ago was, what if we could try and reprogram these not to attack viruses that are infecting bacteria, but what if we can program them to attack mosquito genes or human genes in very specific places? And so this is what happened. So in about 2012, people published some papers that basically boiled down a system that's a little bit more complicated than what I showed you before. But what they showed is that you can actually, in the lab, you can really boil it down to just 2 pieces. So all you need are the molecular scissors, which is this now orange blob in the background, and then you need a short RNA molecule called a short guide RNA, or a synthetic guide RNA, an sgRNA, which is this blue molecule here. And because all it is RNA, we can make that in the lab. Actually, we can just order it from a company now. It's like Amazon Prime. You just go to IDT is the company we use. You say, I want this short guide RNA, and they say, OK, give us $4, and then tomorrow it'll come in a FedEx envelope. And now I've targeted a new mosquito gene. And so we've taken a process that used to take $25,000 in many months, and now we've basically commercialized it and to the point where we can scale this up in a way that we couldn't do before. And so what happens is this short guide RNA, synthetic guide RNA, has the 20 base pair sequence that we want to target because it's exactly complementary to the genome of the animal we're targeting, which is in green. And then it has this loop structure here, and that loop structure is actually what brings along the friend. So the molecular scissors are the same for every experiment. It's one protein that you can grow up and store in vials in the freezer, and you can pull them out whenever you need them. And then the only thing that changes when you want to target a new gene is this 20 base pair sequence here. And so that's the insight that CRISPR has brought, and it's the simplification of the system down to these just two components that has made it so powerful and so flexible, and why it's actually cracking the popular press now in a way that zinc fingers very rarely did. All right, so this is extremely cool. It's kind of an aside, but people have gotten so good with microscopes now that they can actually visualize the Cas9 cutting double-stranded DNA. So the DNA is the kind of wiry structure that you see out there, and then the Cas9 and the guide RNA are in complex in the much bigger structure here. And this is with something called atomic force microscopy. And we're actually visualizing this machine. So the guide RNA has bound the double-stranded DNA here. It's brought along the Cas9, and in the process of about 10 seconds, it actually cuts the DNA. So you can see the two pieces now coming off from that. So this is extremely powerful. This is something that we could dream of for many, many decades, but now we can actually do in a dish or in an organism in a way that is so flexible and so powerful. We're starting to develop that toolkit that the word processor of our dreams had 10 years ago, and CRISPR-Cas is kind of the basis of it today. But you may be thinking, I've told you how to cut a genome. I haven't given you the power to cut and paste and drag and drop and create deletions and all the things that we actually need to do. So we're not clever enough to have figured that out yet. So what we do is we rely on the cells that harbor the genome that we're cutting and say, you deal with it. We're going to make the cut, and then it's your job to actually fix it. But what we can do is we can tweak the way in which that cell fixes it to try and get the effect that we want. So if we want to just make what we call an indel, which is just short for an insertion or a deletion, that turns out to be pretty easy. Because all we have to do is we make the double-strand break, and a cell freaks out when it sees a double-strand break, right? So we have 23 sets of chromosomes, mosquitoes have 3. So what that means is that all 3 billion base pairs are in these very, very long stretches of DNA, and that is the key to how cells can replicate, and the whole process just disappears if you start to get all these double-strand breaks in chromosomes. So the first thing that a cell, a eukaryotic cell at least, does when it sees a double-strand break is it freaks out and it just tries to glue it back together. It's like, let's fix this. We got to fix this. Let's do it right now. And so it takes these enzymes, these ligases that actually just recognize free ends of double-strand DNA, and it's just like, I don't care who you match up with, just fix it. Just jam them back together. And so it does that, but it does that in kind of a sloppy way, because its first goal is just to get that double-strand break closed as quickly as it can. And so what often happens is you will get these small sequences that are deleted or inserted as a result of that process. So it's kind of a sloppy repair mechanism. And so it turns out that genes they have to be in a very specific order. So if you can even shift the frame of a gene, because they encode these short words, these codons of 3 base pairs that then translate to 1 amino acid. So if you can just delete 1 base pair, you have now completely thrown the entire gene out of whack. So it's equivalent to saying, like, if you're trying to read a sentence, and I'm going to delete the second letter of the first word, you can probably deal with that, right? You can see it, and you're like, oh, that's a typo, no problem. But then if what I were to do is to take every letter that comes after that one and shift it over, but maintained all the spaces, at that point, you would quickly start to lose the thread, right? So words would no longer make sense. You couldn't put the sentence back together again if I had just deleted 1 base and then forced everything to move over rather than just letting it be. So that's kind of the equivalent of what we're doing here. But that's still not precise, right? This is kind of a messy thing. It's a good way to break a gene. So if all I want to do is break a gene, Great, that's what we're going to do. We're going to just do this messy repair, let it go back together, and then we'll be good. If I instead want to insert something, so if I want to take a mosquito gene and make it look like a human gene, or if I want to delete one very specific section of a mosquito gene or add something in so we can visualize it, then what we have to do is something that's a little more precise. And we turn to something, again, that the cell already knows how to do, and that's called homology-directed repair. And so if the animal sees a double-strand break like this, and there's another copy of that section of DNA somewhere else in the cell, it can actually use that as a template. And so it does something called strand invasion, where it takes one of these free strands here from the genome, and it's like, okay, well, you kind of look like this, so let me kind of shift this left, shift it right, until we get that double-strand binding just right, and then I'm going to start to copy. And so instead of copying off of this black stuff, which has the double-strand break, it's going to start to copy off of the blue template. And that template could be the other pair of chromosomes that the animal has, or in our experiments, we can actually give it the template. So we can co-inject, we can send along for the ride a short section of DNA that, for the most part, looks like the spot where we made the break, but at one very key region, we might delete a few bases, or we might add in several thousand bases. We can even put proteins that will make the animal glow in the dark. We can just insert them in the middle of this blue thing, and then we've tricked the animal into being like, okay, you kind of look the same on the arms. We call them homology arms because they are identical to the sequence in the genome. And then it's just going to copy through. And when it gets to the new stuff, it has no way of knowing it's new stuff. It just blindly copies through because that's what it knows how to do. And so if we can successfully hijack this homology-directed repair process, this is where the precise part of precise genome editing comes in. So this is where we get to say, you used to look like this, and now I want you to look like this very specific version of the gene. We create that version of the gene, and then we trick the cell into replacing the old version with the new version. So that's really where the precision comes in. And I wish I could tell you that this happened 100% of the time. It doesn't. And so this is one of the reasons that we're not deploying this on a regular basis in the clinic, because it's not good enough if we can only edit 3% of the cancer cells. We have to edit 100% or 99% at least of the cancer cells. And so we can do this, we can do the messy thing with very high efficiency, but we cannot yet do the precise part with very high efficiency. But people are actively kind of working to make that happen. And so that might matter for the clinic, where you have to have 100% efficiency, but in the lab, if we're working on something like a model organism, like zebrafish or mosquitoes or fruit flies, or if we work on bacteria, it turns out not to be a showstopper. Because even if it only works 1 in 100 or 1 in 200 times, we can just select for that 1 or that 2% of the ones that have the correct modification, throw out the rest, and then we go on with our day. Because once we've made that modification, it's there to stay. Because if we do it right, we can put it in the germline of that mosquito, and then every one of the offspring from that edited mosquito is going to carry the same exact manipulation. And so that's what we're trying to do. And so it's okay for us to throw out 98 mosquitoes if we can find the 2 that have the right mutation, but you can't do that with human cells. And so that's why you will see some treatments based on CRISPR that are being developed in things like bacteria or human cells in culture, but we're still a few years off from actually putting those back into people in most cases. All right. So at this stage, let's see, that's about halfway through. So I wanted to stop here before we talk about how we use this in mosquitoes, and maybe see if you guys have any questions about what I've just told you about the mechanisms of CRISPR in general. So does anybody have any questions? Yeah. Here, wait, we'll do the microphone thing too, so people can hear.
It's more of a comment, but
yep.
There's a company called Odin that has produced a at-home or at school do-it-yourself version of this, and it's really cheap. It's like $100 at most.
Yep. So actually, I meant to ask before I went into this whole thing. So how many of you before today had heard of CRISPR? All right, so that's probably half the room, right? So if we had asked this a year ago, 2 years ago, what do you think, Jeannie? 10%? Not even. So this really has only come into our thinking in the last 5 years, and it's amazing to me that half of you have already heard about this. So the next question is, has anybody actually used CRISPR with their own hands? Yeah? No? Was that a hand or no? No? Yeah? No, okay. I saw some movement, and I was almost expecting 1 or 2. There's one up there, so let's give you a microphone, and you can tell me about your experience.
So we are trying it out. I work at Stony Brook University in the summer, and we're trying it out in our biotechnology summer camp.
Yep.
Using plasmids and bacteria, trying it in yeast.
Yep. So if I were to ask, if I come back in a year or 2 years and I ask the same question, I think 100% of you will have heard of it, and probably 10% of you will have actually done it. And I bet if we come back in 5 years, it's going to shift to the point where I would bet that in a decade, most high school biology classes will have a hands-on component where you're actually taking bacteria and you're using CRISPR to make them glow green. Or they actually sell kits now, so if you brew your own beer, you can take the yeast that's in beer and you can edit it with CRISPR to make it glow green under a black light. So then when you brew your beer and you take your friends and you're like, here, drink my beer, then you turn on the black light, it glows green, and they freak out and drop it and think that you've infected them with something crazy. So it's absolutely true that you can go, in my day, we would order sharks or frogs and dissect them. And you had these catalogs of all these animals. They would come fixed, and you'd dissect them and try and learn something about anatomy that way. In 10 years, you guys are going to be doing that with molecular biology and with CRISPR and be editing all sorts of things at the bench, because it really is that easy and that cheap. So that's a great kind of plug for the future. And if you go to college and take biology classes, molecular biology classes with the lab component, you are undoubtedly going to be doing this within the first semester. So that said, any questions about the mechanisms?
I keep seeing PAM sequence, and it looks like it has to be in all the targets.
Yep.
Like, what does it do, and does it have to be there?
So that is, I tried to skip over it, but there's no skipping over it with you guys, which is great. So the PAM sequence is that little yellow bit right here. And so it's called the protospacer adjacent motif. And so let's see if we can kind of come up with why that has to be there on our own. I'll try and talk you through it. Lost my clicker. All right. So I told you that this whole thing is an immune system, right? So it sees the phage genome, it cuts it up, and then it puts it into its own genome, and then it uses that as a template to kind of cut up the phage the next time it comes in. So why would this system not work if it worked? If it was just that simple, what's one of the problems that you could see right off the bat? So we're just using 20 base pairs, and we're saying, I'm going to copy that 20 base pairs from virus to bacteria, and then the next time the virus comes, I'm going to use those 20 base pairs to recognize the virus and cut that up. Yep. Yeah, there might be a repeat of those base pairs. That's absolutely true, but it's worse than that. The virus could mutate, sure, that's another problem. So that's why we need to make lots of these. So just like our body makes more than one antibody, we're going to make more than one of these. You were close. So the first one about there being another copy is the right answer here. Anybody want to take a stab?
The bacteria could recognize that itself and then destroy itself?
Exactly. So that is exactly what would happen if it were really that simple, right? Because it would take this green sequence, it would make this little CRISPR RNA here, and then all that RNA is doing is looking for the DNA copy of itself. And so if it finds that in the bacterial genome, which is where it came from, it would just make that double-strand break. And now the bacteria has the same problem that we're trying to induce in the virus. It would have this double-strand break, and it would freak out, and the whole system wouldn't work. So instead, what it comes up with is this extremely clever way that we call the PAM, where it basically says, we're going to take these 20 base pairs, but in addition to that, we're going to require that in the thing we're cutting, there is this sequence, this protospacer adjacent motif called a PAM, which for the one that we use every day is just an NGG. So N stands for any base, so really it's just a GG sequence. And that has to be in the target DNA, but is not in the guide RNA itself. So it is here as part of the thing that we're trying to cut, but it's not actually present in the guide RNA molecule itself. And so what that does is it lets the bacteria make all of these templates, produce all of these RNAs, but because it does not have this protospacer adjacent motif in the bacterial genome, the molecular scissors, which are the big, the Cas9 blob shown here, it's not going to see this PAM. And there's actually evidence to show that it first has to find the PAM, and then it checks to see if the rest of it matches. So it needs both of those things to match before it will make the double-strand break. So by having this requirement for a protospatial adjacent motif, you're protecting the bacterium from cutting itself, and that's really the way that this whole thing can work as an immune system to target foreign DNA and not your own genome. So that was a perfect question. So we'll come back to this at the end, so if anybody has questions towards the end about CRISPR, we can go back to that, but I do want to keep moving so we can talk a little bit about the mosquito work before we get there. All right, so how do we use CRISPR to fight mosquitoes? The first thing we have to do is generate stable and transmissible mutations. So if we want to release mosquitoes, we need to mutate them first. That's one possibility we'll talk about. The other is if we're in the lab and we want to study CO2 or heat, we have to make those mutant mosquitoes, and then we have to propagate them, right? We have to have a strain of mosquito that always has our mutation, because we don't want to be, we work with thousands of animals, and we can't induce that same mutation every time. We want to get it into their germline. So how do we get the mutations into the germline? So we came up with a protocol a few years back that just takes these two components, so this Cas9, the big blob, and the guide RNA, and we actually inject that into a mosquito embryo, which is about the size of a poppy seed or a sesame seed. So it's the eggs that the mosquitoes lay. We wait about half an hour or so they can kind of settle in a little bit. And then we actually just take a glass needle and we inject the protein and the RNA into the embryo itself. And so then what we're going to do is we're going to make that double-strand break. We're going to get all of these insertions, deletions, and repairs. But it's not going to be a stable germline mutation at that point. So what we have to do is we have to do this in just the right way so we target one side of the embryo where we know that the germline is going to develop. So we can't sex the embryos. I can't inject one embryo and say that's a female, that's a male, that's a female, that's a male. But what we do know is that the posterior pole of the embryo is where both the sperm in the males and the eggs in the females are going to develop. And so what we're doing is we're trying to edit the precursor cells that are going to make sperm and eggs. So then if we get that right mutation, it's going to be present in their germline, and then we will look at their offspring and find the ones that have carried on that mutation. And if we can do that, then we're in business, because then we've made an animal in which that mutation is present in every cell in the animal, and we'll pass it on to its offspring as well. So this is a video of this in process. This was done, again, by Connor McMenamin, and they're injecting zinc fingers at this point, but it's the same exact process for CRISPR. And so you can see these are the mosquito embryos lined up on a glass slide. This is a very fine glass needle, and you can see them it has a very sharp tip, but the eggs have this kind of chorion, the shell on them, so you have to puncture the chorion, and then actually we're just injecting liquid into the eggs, and that liquid contains the protein and the guide RNA. So this is still a very physical thing. We have to physically inject these proteins and RNAs into the embryos. There are other ways to if you're working with human stem cells, for example, you can do this with different methods. But whatever organism you're working on, you have to find a way to introduce these reagents into the animal. And we do it by injecting eggs. All right, so we use CRISPR, I told you before, because it's much more flexible and easy to use at the bench than zinc fingers or talons. So that's why we pick it. But that said, all of the work that Leslie told you about earlier was done using zinc fingers, and those are absolutely viable tools. They're just a little bit less flexible and harder to work with. All right, so why would we want to do this in mosquitoes? So we'll talk about trying to make mosquitoes go extinct in a second, but when we're doing basic science in the lab, we just need to have these stable mutations so that we can understand the genetic basis of all of the beautiful behaviors that Leslie introduced you to. So if we want to know what receptors they're using to smell CO2 or what receptors they're using to smell humans, we can make some guesses by sequencing the genome, but for certain things, we might only be able to narrow it down to a list of, say, 10 or 12 proteins or genes. And then we don't know which of those 12 it's going to be, or maybe it's going to be a combination of 2 or 3 of those 12 if they're subunits of a certain receptor. So we need to be able to mutate them and then ask the question, can these animals still host-seek? Can they find and bite a human being? Can they still infect themselves and then transmit viruses like dengue or yellow fever? Can they find, so this is something that I've been working on, can they find appropriate bodies of water, so standing water for their eggs to be laid into so their offspring can develop? So if our mosquitoes make a mistake and lay their eggs in salt water, all of their larvae are going to die. All 100 of those eggs are going to hatch, and then all of the larvae are going to die. So it's an extremely important decision point for the female mosquito to decide where she's going to put those eggs, because then they spend about a week on their own, and there's no parental care. It's not like a bird where she's going to go find them food, bring it back. So she has to find just the right place so that as many of her offspring can survive as possible. So these are some of the reasons that we make these mutations in the lab, because we want to do basic science and we need to be able to test our hypotheses in the lab. But maybe something that you've heard about, we'll talk about in a second, is something like gene drive. And there people are actually talking about making genetically modified mosquitoes and releasing them into the wild. And so to do that, we're going to have to jump through a lot of kind of regulatory hoops. But I'll just show you a few examples of what we do. So this is a genetically modified mosquito. She looks normal, she takes blood, but she has a mutation in a receptor that we think helps them find clean water in which to lay their eggs. So the females aren't as good as discriminating a site where their offspring will thrive and be happy versus one where there's enough salt in the water that the offspring will die. So to the naked eye, these look like normal mosquitoes, but when you actually put them under a microscope, it turns out that we've made the manipulation in just the right way so that they're expressing these fluorescent proteins that make them glow blue or red under the right wavelength of light. And so this, in this particular case, is just how we track the mutation. So it's kind of a side effect of I broke the gene that I'm interested in, but at the same time, I made the animal glow in the dark because that makes them easier to find. And so that experiment that Leslie talked about with the CO2 mutants, where you release them into a cage and then you want to figure out which ones end up finding and targeting humans, they were also done like this, so that you could just put them under a microscope and say wild type, mutant, wild type, mutant, instead of having to do molecular genotyping on them. So a lot of times we'll do the repair just to make our lives easier, so that I know I have my blue mosquitoes and my red mosquitoes, and if they ever get mixed up, I can just kind of sort them out and be like, okay, blue's over here, red's over here. But we can actually do more complicated things, where this is now the very tip of the proboscis, which is the part of the female that's actually contacting the skin. It's not the syringe, but it's the part of the female's anatomy that sits on top of the skin while the syringe is puncturing and then sucking blood. And so now what we've done is we've labeled a very specific set of sensory neurons with a red fluorescent protein, and you can see them here. And so these are sensory neurons that we think are responding again to things like water and salts, so that the animals are not only sucking your blood, but they're tasting your skin at the same time. And so both on the tips of their proboscis, but then also on the tips of their legs. So this is the leg of the female mosquito, and these are the same kind of molecular class of sensory neurons, but now they just have this fluorescent protein that shows me exactly where they are, so I can go in and study those cells in more detail. And you can see that some of these actually kind of innervate up near the claw of the mosquito, and that's what they're using to kind of, we think they're tasting with their feet, basically. And so this is a way that we can study the cells, the taste cells that they're using to sample their environment. So this is what we do in the lab. What you may have heard more about is people talking about genetically modified mosquitoes as a potential control tool. And to do that, you have to talk about whether we're going to genetically modify mosquitoes and then release them into the wild. And so I'm going to spend just the last few minutes before questions on that, and then we'll get into your questions. But in order to influence a wild population of animals with a laboratory-generated strain, what do we have to do? We have to release the laboratory-generated strain. And then that laboratory-generated strain has to be able to compete in the wild. It can't be this kind of like if you take a tiger that's raised in the zoo and then you take it and release it out in the wild, that's probably not going to be as efficient as an animal that actually grew up and learned all the behaviors it has to do in order to survive. So we have a similar thing here where we have to make sure that the modified mosquitoes are fit, as we call them. But they can't just be fit, because we're not trying to replace wild mosquitoes with mosquitoes that are red under a microscope just for the fun of it, right? That's not going to do anything. But what we're going to do is we're going to release them, and they're going to have very specific deficits. So we might release males that are sterile. And why would we do that? It's because the males would then mate with a female, but they would not be able to fertilize the eggs. The females would still think they've mated, and they would reject future male copulation attempts, but then they wouldn't be able to lay any viable eggs. And so that is one thing that we're actually doing now is we, as the scientific community, have developed sterile males and are releasing them into very restricted geographical locations to try and knock down the population of Aedes aegypti. And then there are some more complicated strategies that people have been imagining, things like gene drive, which I'll touch on really briefly, which in theory, if we do this right and it's efficient and the animals don't evolve resistance, we could make entire species go extinct with something like gene drive. And because we're breaking the rules of inheritance, and we're actually, they call it super Mendelian inheritance. you can engineer animals so that they're not only passing on one copy of a gene, but they're always passing on 2 copies of a mutation, and then that will spread throughout the population. And eventually, if you do it right, that can cause an entire species, either locally or globally, to go extinct. And so that's the ultimate goal of some of these gene drives. And then there's some kind of middle of the road tools. So maybe we don't release sterile males, maybe we don't try and make a population go extinct, but maybe what we're trying to do is to spread a transgene that makes an animal resistant to something like malaria or dengue or yellow fever. And so this would be altering the phenotype of the animal, not to kill them, but to make them harmless, essentially. So we could make them so that they could even still bite me, but if they're not capable of transmitting virus, they're going to be much, much less of a worry for public health and other areas. All right, so the sterile males are fairly simple. The concept is fairly simple. I talked about it before. This is the life cycle of a mosquito. A male has to mate with a female, but if that male is sterile, meaning he'll go through the whole mating process but then won't fertilize any of the eggs, you can essentially cut off any of the offspring that result from this mating. But this is going to be a problem because it's not going to be stable, right? So what we can do is we can release a ton of males, but then once they've mated and died in a month or 2 months, we're still going to have all of the wild-type mosquitoes that escaped. So maybe there were still some wild-type males, so some version of the wild-type matings happened. Maybe we knocked the population down for a while, but then there are still these wild-type animals, and if we don't release for a while, then the population levels are going to go right back to where they were. So this has been done on the local level. I said earlier in South America, so this is the man releasing these sterile males. It's a company called Oxitec that has developed a strain, OX513A, which they've put through all the testing and regulators have approved the release. And so what they'll do is they'll drive around on trucks sometimes and just release cages of tens of thousands of these mosquitoes into areas where there's endemic dengue fever or Zika. And so that will locally cause the populations to dip for a while. And then if you time it right and you get the right geography, you can actually reduce instances of disease transmission through release programs like this. This is a flyer that Oxitec put together when they were trying to convince people in Key West to release these mosquitoes there. And it turns out that people in Key West weren't too happy about the idea of somebody coming through and releasing tens of thousands of mosquitoes that have been genetically modified in a lab. So this is their propaganda. I tend to side with them because I'm a scientist and I think it's right. But the type of arguments you'll see on the other side are similar to the same types of arguments from people who don't want to eat genetically modified produce, right? So in Europe, in a lot of countries in Europe and in some parts of the States, there are requirements that you label any food that's been genetically modified because people are either scared of the implications of that or they have legitimate scientific worries about what this does to the nutritional value or what's it doing to the wild crops that haven't been genetically modified and all of that. So there's a lot of pushback in the public sphere from people who have both what I consider legitimate concerns and then more kind of scaremongery techniques about the possibilities. So this is happening in our country now. The FDA has cleared these for release, and now they're just in the process of trying to find communities that are willing, essentially, to go through with these release programs. And they've had some pretty striking success. So they can reduce populations by up to 96% in areas that they've released these mosquitoes in South America. So this could be an extremely powerful control tool. The gene drive is kind of the one that is a little bit scarier in the sense that if you take some of the simple systems, you're trying to make a species go extinct. And so when you hear gene drive, like that's the logical extreme of what people are talking about. And so the way it works is that normally you would release an animal that has a transgene, and only in half of their offspring would you get the propagation of that transgene. So eventually it would just kind of peter out, right? You would have to release many, many, many more mosquitoes than were already present in the local ecology if you actually wanted to replace a whole gene with your new copy. What gene drive does is it breaks the rules of normal inheritance in a way that involves the CRISPR-Cas type of systems. It actually uses CRISPR to cut its own chromosomes and takes a heterozygous animal, so an animal that only has one copy of a transgene, and then it forces, it basically makes the wild-type copy of that same gene to take on the mutant phenotype by doing a local rearrangement inside the cells of the animal itself. So it's cutting the wild type copy of the chromosome and then copying over the drive system, the entire system, into the wild type chromosome. So then you have an animal that would normally be heterozygous has now converted itself into a homozygote, and it's going to pass on that drive to all of its offspring. And the drive, if it doesn't evolve some kind of drive breaking mutation, is going to eventually propagate to every single animal in the population. And so if there are unintended side effects with this sort of procedure, it's hard to imagine going back from a scenario where you've had a successful drive. And people say, well, it's okay, we developed one drive the first time around, so we'll just develop another drive to reverse what we did the first time and then release that into the wild. And that's where you get into this kind of Jurassic Park kind of scenario that people are talking about. So, all right. This is just a slide. I'm not going to go through it, but I want to make it clear that people, when they say gene drive, they can mean a lot of different things. So they can either try and make sterile animals. They can try and make it so that offspring will only be male or only be female, and that will eventually cause populations to decrease. So people are thinking about this, and they're thinking about things like how hard is it going to be to actually build the drive? What's the risk of it spreading? What's the ecological risk? So for some of these, you can see that people are saying possible extinction, precipitous decline with possible extinction. And then for some of them, they may revert over time, so they may be kind of time limited, so they're not going to have that kind of long-lasting effect. But there are a lot of different variants on this. And then there's one that doesn't involve making the animals go extinct. And this is one of the malaria vectors, so it's Anopheles defensi. And they've actually engineered a drive system. So it has the CRISPR-Cas components here, which are what it's going to use to copy itself from one chromosome to another. But then along for the ride are these two proteins over here called M2A10 and M1C3. And what these are transgenes that the animal wouldn't normally express. But if it does express those genes, they become resistant to malaria infection. So you're basically giving this mosquito a jump start in terms of its immune response to malaria. So they're called nanobodies, and they're basically very short protein antibodies against malaria. And so this drive system, if it were to get released into the wild and work to perfect 100% efficiency, every Anopheles defensi mosquito that has been affected by the drive would now be producing these nanobodies that would make it more resistant to malaria. So this is kind of one of the middle-of-the-road strategies that people are thinking about. All right, so I was going to ask some questions and give away some stuff. And then I want to hear your questions. But let's start with the easy ones. So who remembers how many base pairs the human genome is made up of? The front row. All right, you get your thing. But for those of you who have taken notes for the next ones, don't look at your notes, and we'll see if this actually stuck. So how big is the mosquito genome? You can give me the order of magnitude, not the... What's that? Billion. All right. Billion is the way to go. CRISPR is what in bacteria and archaea? An immune system. Very good. These are the quick ones, and then we'll go to the microphones for the real ones. All right. What are the two repair pathways? Does anybody remember what the messy repair pathway is called after we make the double-strand break? Non-homologous end joining. That was a good pull. Do you want malaria, do you want Zika, or do you want a mosquito? You'll have Zika. Nice. Congratulations, you now have Zika. All right, anybody remember the precise repair pathway for either a mosquito or malaria? I'll give it to you. Do you want the mosquito or do you want malaria? Malaria. So homology-directed repair was the answer to that one. So these are just some of the simple concepts. So I'll reveal the answers. We all did well on that. And now for the final, somebody gets the mosquito. No notes, and you're going to have to do it to a microphone. All right.
Clustered regularly.
All right, quiet down. We're going to get... This is spelling bee style. So you got to give me the acronym, what each of the letters stands for. Go for it.
Clustered regularly. Interperspected.
We got halfway there. So we got
Interspaced.
clustered, regularly interspersed.
Interspersed.
And then what's the P?
Short.
Yep, short is good. Anybody want to help her out with the P? Palindromic.
Palindromic.
And then,
Repeat.
well done. Round of applause for that. Nice job. And then for the last one that's slightly easier, we'll go over there. I'll let the microphone holder pick somebody. How about right there? Go for it.
CRISPR associated system.
Well done. Bravo. That one, once you get CRISPR, that one's straightforward. So, all right, with that, I will take your questions. If you want to talk about CRISPR or mosquitoes or anything else, I think we have a few minutes, so go for it. All right, yeah, we'll start up there, and then maybe if you want to give somebody a microphone, we'll go to the left next. So go for it.
You brought up introducing sterile males as like a way to like control the population. But if a female goes through that cycle of reproduction twice in her lifetime, and she like gets a male the first time around that is sterile, will she recognize that and the second time around like try and find a new mate?
That's a great question. So the question is, if a female can learn that they've mated with a sterile male the first time around and then try and avoid a similar type of male the next time. So I would say we don't have evidence that they can kind of actively learn that behavior, but there is evidence that females can choose males based on something like the flight tone, the frequency that they beat their wings, and there might be some kind of active like call and response going on there. So I would say it's more of an issue if the sterile males that have been generated in the lab, and they're more like the zoo animals, if they're not as good at competing for females with the wild type males, then they may just be less efficient at the competition, and then the sterility wouldn't have the same impact that it would. So we don't have evidence that the females learn, but we do have to make sure in that instance that the males are fit and they can compete with the wild type males. So, all right, let's go over there.
Do you personally have any ethical dilemmas when conducting these tests as it relates to humans?
So ethical dilemmas as it relates to humans. So using CRISPR, you mean to edit human genomes? Absolutely. I have many, many, many ethical questions about that, and I'm kind of glad I work on mosquitoes and don't have to think about it. But so I told you how you could make sterile, heritable mutations in mosquitoes, but you're absolutely right that most of what you'll read about today in the popular press is people talking about using CRISPR to make heritable mutations in humans, right? So in theory, if you knew a gene that controlled a disease, so if there's a monogenic disease like Huntington's disease, and you could test for it, and then you could actually remove the repeats, the Huntington repeats, from a woman, and then she could have offspring that would not carry those repeats, in theory, those offspring would now not have Huntington's disease, right? So that's the medical application. The other one would be, you could think of if you know the genes for hair color, or you know the genes for intelligence, or you know the genes for athletic ability. In theory, you can now introduce the copies of those genes, the versions of those genes, into a woman or a man's germline, so then all of their offspring would carry that trait. So there is a huge scale of the types of things that you can do. And personally, I think everybody's going to have their own opinion, and experts definitely have differing opinions on this. But personally, I think the power of it for addressing certain types of disease is too powerful to ignore. And if it has the possibility to do good, we have to investigate that. But I do worry about it going too far. And if we get into a situation where people are using it for things that aren't directly medically relevant, then that's where I personally would start to have issue with it. But there are dozens and dozens of scenarios you can come up with that would raise all sorts of ethical questions. So it's a great question. Let's go over there, and then we'll get you a microphone. So anybody have a microphone up there? Yep.
You talked about the company that released mosquitoes in South America.
Yep.
Did anybody look at, that wasn't a gene drive, right?
That was not a gene drive. So those are sterile males, and the company is called Oxitec. Yep.
Did anyone look at the ecological impact of that, or at like how far the mosquitoes were able to spread, or how long they were around for?
So I know for the second part, they absolutely looked at how far they could spread. And I don't remember the exact distance, but they were trapping. So in order to know how efficient it is, you have to set up traps and then see how many mosquitoes you catch, and then you can also genotype them and ask if they have the drive or not. And so they know how far it spread geographically, and the answer is not that far. I don't remember the exact distance, but these mosquitoes fly probably hundreds of meters in their lifetime, maybe a few kilometers, so they're not migrating or anything like that. So with something like a sterile male where it's restricted to the animals that you release, that's going to be pretty geographically restricted. The question about the ecological impact, I'm not sure if they looked at that. This was not designed to make the species go extinct, so it's a little bit less of a concern. But one thing that I've heard anecdote about is in areas where they have knocked down the Aedes aegypti population via mechanisms like this, you will start to see an increase in other species of mosquitoes, right? And so this is actually something that's extremely important with malaria because there's not just one malaria mosquito. In some places, there are up to 14 species that can transmit malaria. And all of the gene drives and genetically modified mosquitoes are going to be restricted to one of those species. So if we just, for example, cause Anopheles gambiae to go extinct, which is the major malaria vector, we might just be creating an ecosystem where Anopheles stephensi, or one of the other malaria mosquitoes, now has free reign because they're not having to compete locally. So I would say that's one of the ecological issues to think about, but as far as I know, they haven't studied that with the gene drive or the sterile males. So there was one over there.
I was wondering, in terms of controlling mosquito population, has anyone found or been interested in finding natural variants of mosquitoes that are resistant to disease, expanding the population and releasing them? And then also, are you allowed to do gain-of-function experiments on mosquitoes that might be more advantageous in terms of their reproduction so that they can easily enter the populations?
So I'll answer the second part first. We are absolutely allowed to do that in the lab. So we can do anything we want with limits, some limits, in the lab. So the gain of function in terms of making them more fit or more able to pass on their genes, we can absolutely do that. That's what the nanobody gene drive is for the malaria mosquito, is essentially a gain of function. It's now expressing these nanobodies against malaria. So as far as I know, they haven't gotten permission to release these yet, but they've done field cage trials in kind of environments that are mimicking the natural environment. So there's going to have to be regulation involved with the release of any gene drive, and I would bet that whether or not there's some sort of gain of function transgene in there is going to be looked at as one of those. Because it's a little bit different than making a mosquito that's worse at doing something, because that may just cause its numbers to fizzle out. But if you're trying to make a super mosquito that, for example, likes cows but not humans, and then trying to replace that, you can imagine all sorts of ways in which that would go wrong. So I imagine that the ethics are going to get more complicated if you try and have gain of function. But as far as I know, none of the gene drive systems have gone through all of the regulatory process to actually be released in the field yet. But people are talking about it in the next few years. So, all right, got one in the second row.
You talked about before about how large the amoeba genome was. What's like a specific reason for that?
I have no idea. And it's actually, so it takes a lot of energy to copy a genome, right? And so you would imagine that there would be selection pressure on making that genome as small as possible. And there's a lot of evidence in viruses, for example, that because they have to inject their genomes into hosts and they have to very quickly propagate, that there's huge selection pressure to make the genomes as small as possible. And so you'll even have genes that you can read in one direction and produce one protein and read in the other direction that produce a different protein. And so they've come up with all sorts of clever tricks. I have no idea why you would want it to be large. I suspect it's just kind of an evolutionary accident, and they haven't evolved to get that back down yet. So a lot of times genomes will evolve by duplicating themselves and then like playing with some of the different copies. And so if you've caught them in an evolutionary time point where they haven't trimmed out all the fat, basically, you might get there. But who knows, there may be a reason they want it to be that big, and I don't know. All right, someone over there with a microphone? Go for it.
This is more like a question on your opinion about like what you feel like would be the most like the best way to go about using CRISPR, and just in terms of mosquitoes for right now. Like how would you think would most benefit the environment or like whatever the diseases?
That's a great question. Personally, I think it has more use in the lab right now. So it's allowed us to do experiments that we couldn't have done 5 or 10 years ago in terms of the scope and scale of the numbers of genes we can edit and the manipulations we can make. So I personally think that the biggest benefits, both for mosquitoes and also for human health, are going to be in its applications in the lab that allow us to understand these systems at a much more detailed level than we could before. And that's going to let us do things. So there are many, many ways to control mosquitoes that don't involve releasing gene drives, right? We can develop new repellents or new insecticides so that in areas where they've developed resistance to a pyrethroid, and that's become a problem, we can now understand the mechanisms by which a new insecticide might work, develop that as a chemical, and hopefully make it safe so that you can use it widely, and that Jones Beach picture may, again, be a reality. But to do that, we have to understand the biology of both the sensory systems and the physiology that allow that insecticide to work. So you hear a lot, especially in magazines and the popular press, about the use of CRISPR to make designer babies and to make gene drives to make things go extinct. George Church at Harvard wants to resurrect the woolly mammoth. I don't know if you've heard about this. Has anybody heard about this? He wants to take a species that's now extinct and basically resurrect it using things like CRISPR and synthetic biology. I think that's that's out on the event horizon a little bit further, but maybe not that far away. But it has definitely transformed our ability in the lab to understand mosquitoes and all sorts of different systems. So, all right, got one up there. And then Sean's given me the cut, so last question.
Are there any NIH guidelines for using CRISPR?
Yes. There are NIH guidelines, there are NSF guidelines. None of them, as far as I know, are legally binding yet. So there have been, both in the US and abroad, a lot of meetings convened, especially around the use of CRISPR in human experiments. And there was a proposed moratorium on developing germline edited humans that a lot of people signed on to, and then some scientists who were in China went out and did the experiment anyway. And so I think it's sort of at this point now where there are guidelines, and maybe you can't get funding for certain things from the NIH, but you can always find private funding, or you can move to a different country to get around specific guidelines. So it's a little bit of a free-for-all right now. But there are absolutely regulators and scientists who are thinking about ways to put some ethical boundaries on this. So great question.
I have a couple of these left over, so I want to ask one quick trivia question back to you guys. Ben is using the CRISPR-Cas9. It's sort of like a full circle situation. Ben was using a CRISPR-Cas9 system from a bacterium called Streptococcus pyogenes. Does anybody, and that caused a disease which basically made Rockefeller a reality. Does anybody remember what that disease was? What do we have? Thanks, everybody. All right, so it was such a great day. I am so glad everybody was able to join us today. I want to take a few minutes to acknowledge all of the people. Thanks, everybody, for quieting your friends down. For who made this possible. First and foremost, Sean Davis and Kathleen Gaffney. You guys killed it. Thank you so much for all of this. We have Merck to thank for, again, in their support of this year's Talking Science. And we have tons of people working from the development team. RockEDU specifically, I just want to give a shout out to Odelis, who just helped so much with putting the activities together in the CRC. We have the IT team. Thank you, Izzy, for being up there. Jeff behind the camera. We have Todd over in Caspary. Thank you for all the work that you're putting into this. Our teams with custodial, our teams with catering, they're all amazing. And last but not least, thank you to Ben and Leslie, who took the time. I mean, these are amazing, wonderful scientists giving back to the community. So thank you so much for the efforts that you're putting in today. And thank you to the teachers. Holy cow, you guys are at the front lines of education. Without a good teacher, there would be no scientists. So thank you. And to you, to all the students, your questions were amazing. Your dedication is amazing. I hope that you'll continue checking us out at RockEDU. We have lots of programs outside of Talking Science, which happens annually. RockEDU Presents is happening on January 19 if you want to look at our website to see our next lecture series coming up. And keep in touch and safe travels home. Thanks again for being here.